ninth virtual class in which we are going to learn and understand about the topic called translation under the chapter Molecular Basis of Inheritance. This is one of the most important events which occurs in all the cells for the metabolic activities in which the proteins will be synthesized. The process of translation is most vital, which results in the synthesis of various forms of protein in the cell, which ultimately plays a role for the metabolic activities. The translation is a process of polymerization of amino acids to form polypeptide chain that is acting as a protein. The order and the sequence of amino acids to form a polypeptide chain is defined by the nitrogen bases present on the mRNA in the form of codons. So it is the coded form of information present on mRNA that will give the order and the sequence of amino acids in each polypeptide chain of amino acids. So the whole process of polymerization of amino acids to form a polypeptide chain is described under 4 major headings. So before going to that, let me repeat, the process of translation is called as a protein synthesis, in which the polymerization of amino acids takes place, which results the formation of polypeptide chain. The whole process of translation is carried out in the presence of various enzymes like amino acid synthetase, peptidal transferase, releasing factor, and each and every amino acid which take part in the process of protein synthesis is going to be get activated by the ATP, which is commonly called the energy currency of the cell, which is going to guide and also activate the each amino acid to take part in the process of protein synthesis. The whole process of translation is studied under 4 major headings. One is activation of amino acids. Then second one, initiation of polypeptide chain. Then third one, elongation of polypeptide chain, and lastly the termination of polypeptide chain. This process of translation is in a sequential order where there are number of amino acids are involved. As we know, in each cell there are 20 different types of amino acids are present. Each of this amino acid will get involved in the process of polymerization of amino acids to form polypeptide chain. For that, each of the amino acid will get activated first by ATP. After activation, each activated amino acid will get attached to tRNA with the help of some enzymes like amino acid synthetase enzyme. So that makes the complex of the amino acid which is ready for the process of translation. So to carry out this entire process of translation, along with this 20 amino acid, tRNA, one of the form of RNA which involves, then mRNA called messenger RNA, and the number of ribosomes which associated with the process of translation, and several enzymes will work for the process of translation. So the whole process of translation ultimately gives the product called protein. As we know, protein is a body builder. Every cell needs one or the other type of an enzyme, one or the other type of an hormone for the metabolic reactions. Here almost all the enzymes and hormones are made with one or the other form of a protein, and the protein synthesis is carried out inside the cell, that is in the cytoplasm of the cell under the influence of DNA, which passes its information from DNA to RNA. As we know, the RNA received the information from DNA in the form of an codon, and that coded form of information which is present in an mRNA is going to play a role for the process of protein synthesis, which decides or which gives order and sequence of amino acids to form the polypeptide chain. One of the most important events called translation. So as I mentioned, it will be going to occur in 4 different steps. Activation of amino acids, the first step in which each and every amino acid will get activated. Let me describe the first one. Each amino acid combines with ATP, adenosine triphosphate, called energy currency of the cell, plus amino acid synthetase enzyme. This will make an amino acid to become active amino acid. So this is inactive amino acid. After this attachment, it will convert it into active amino acid. This active amino acid will combine with tRNA and it will form tRNA will combine with active amino acid to form an amino acid tRNA complex. This is one of the most important event where each and every amino acid will get activated in the very first stage of translation, because all these 20 amino acids which are present scattered in the cytoplasm, they are found in an inactive form. All inactive forms of amino acids will get activated with several enzymes and ATP, resulting amino acid will become active. The active amino acid is going to work for the process of the translation, where initiation of polypeptide chain takes place. This I will explain you with the help of a chart here. The process of translation begins with the first step, activation, as we have just now discussed, in which amino acids will get activated. Now each active amino acid combined with tRNA and it forms a complex called amino acid tRNA complex. This complex proceeds towards the mRNA. Remember, here mRNA is acting as a template. The mRNA acting as a template which possesses the number of codons, that is coded information present on mRNA. Units of ribosomes also plays an important role here. So after activation of amino acid, the next step is initiation of polypeptide chain. For that, or at the time of initiation, the smaller subunit of ribosome comes and attaches to the mRNA at 5' end. And at that stage, one of the tRNA complex comes and attaches to the codon, that is at AUG. As it will comes and attaches to AUG, that time the larger subunit of ribosome, which is having the three sites, A site, P site and E site, will comes and attaches to the smaller subunit of ribosome. I'll repeat. First, the smaller subunit comes and attaches to the mRNA here. As it will attach, that time the tRNA complex with amino acid, the first amino acid, comes and attaching to the codon called AUG. Remember, AUG is called initiator codon, one which initiates the process of protein synthesis. So it is the AUG which is acting as an initiator codon. At that stage, one tRNA complex carrying the first amino acid comes and attaching to the smaller subunit of the mRNA here. At that stage, the larger subunit comes and binded with the smaller subunit, and here the ribosome is acting as a catalyst to form the bonding between the two amino acids. Okay, so in the beginning, the initiation of polypeptide chain, the two ribosomal subunits attaches, and the first tRNA which is happened to carry the methionine, the first amino acid called methionine, has been brought to mRNA by tRNA, and it will attach by means of complementary base pairing. As the tRNA complex, which is reaching to the mRNA, carrying the first amino acid called methionine, bearing the anticodon UAC. This UAC will be complementarily base paired with AUG on mRNA, and that will begin the process of peptide chain formation, or nothing but the process of protein synthesis begins. That is what we call initiation of polypeptide chain. Followed to that, the second amino acid attached with second tRNA, like that the third one, fourth one, like that subsequently. Each amino acid will attach with one particular tRNA, and one by one the tRNA bringing that amino acids towards the mRNA. So in the next step, second step, the elongation of polypeptide chain. After beginning, the next is elongation of polypeptide chain. So as the first amino acid is coded by AUG, and it will convert that methionine into functional methionine. Now this functional methionine will going to link with subsequent amino acid which has been brought by tRNA. For that, ribosome subunits which have been attached during initiation will move from codon to codon with the help of an enzyme called peptidal transferase enzyme. As it will move from codon to codon, each time one amino acid has brought by one tRNA. So at that time, the ribosome acts as an enzyme to link two amino acids with the help of peptide bond, and that forms a chain of amino acid called polypeptide chain of amino acid. So here the ribosome will move from codon to codon, and the first codon, after getting activation of the amino acid, it will go out from the ribosome through E site. Now the second tRNA enters to the mRNA. By the time the ribosome will move to second codon, that is move toward the second codon. Now the second amino acid will get coded. At that stage, the first amino acid and the second amino acid will be linked with the help of a bond called peptide bond with the help of ribosome. Like that, it will go on moving from codon to codon for polymerization of amino acids. Order and the sequence of amino acids present in the polypeptide chain is totally found in an mRNA. The order and the sequence of amino acids present in a polypeptide chain, that information is present in the form of codes on mRNA. Like that, one by one, the tRNA will go on reaching, and polymerization will continue to form a long chain of polypeptide chain takes place. The amino acids will be go on linking with each other, and it will form a long chain of amino acid. By the time it will reach us towards the 3' end of mRNA, the ribosomal subunit, which has moved from codon to codon, it will show the long chain of amino acids by the time it will reach us towards the terminal end, towards the 3' end. Now coming to the last step of translation, termination of polypeptide chain. While the ribosomes are moving from codon to codon, it is reaching towards the 3' end. Near 3' end, there is a code called UAA or UAG or UGA. Any one among this code is present near the 3' end, that is playing a role to stop the process. That's why it is commonly called stop codon. Near 3' end, any one among this code will be present near 3' end, while the complex of ribosome with the chain of amino acid reaching towards the 3' end and encounters with any one of this stop codon or terminator codon, there is a release factor which attaches to this complex. As a result of attachment of release factor to the complex, the two subunits of ribosome which are joined with each other, that will dissociate or separates, and the chain of the amino acids will detach from the ribosome. So as the ribosomes will dissociate, amino acid chain will get detached. That brings about the end of protein synthesis or the termination of polypeptide chain. The number of amino acids present in this chain, further it will not going to add any amino acid because of the releasing factor which releases the chain because of the encountering of stop codon or terminator codon, that brings about the end of polypeptide chain formation called termination of polypeptide chain. So these are the 4 steps which are involved in the process of translation. So at the end of polypeptide chain, the polypeptide chain which is released is going to be act as a protein. Further, this chain will undergo modification or some structural changes under some certain organelles of cells like endoplasmic reticulum, Golgi complex, resulting in the formation of particular type of protein or particular type of enzyme. So what kind of enzyme, what kind of protein is needed by the living organism for their metabolism, accordingly it will be modified or it will be designed in Golgi complex and endoplasmic reticulum, resulting it will produce or it will release that enzyme or protein in the cytoplasm of the cell for the metabolic activities. So this whole event of translation is most vital event for every living being. To show metabolic activity, the enzymes or proteins are needed, and that whole process of enzyme or protein which is supplied by means of a process called translation. So translation is the event which supplies the regular proteins for the cells. It is the process of protein synthesis which is guided by DNA by utilizing mRNA and other number of enzymes and different types of RNAs. So in the next class, we're going to discuss about one more very interesting concept called regulation of the genes or gene regulation. When the gene will work, how the gene will work, what are the different activities which are going to occur in a cell which makes the gene to work, in which circumstances gene will work, in which circumstances gene will not work, when gene will work, when gene will stop working. That is what we call the gene regulation. One very interesting and beautiful concept we'll discuss in the next class. Till then, stay at home, be safe. Thank you.